So the next presentation is LaTonya Williams. Presentation title is Mechanisms of HIV-1-Specific IgG3 Effector Function and Protection Against HIV-1 Infection. Dr. Williams is also a T32 recipient.
Thank you. Good morning. I'd like to first thank the CFAR for the invitation and the opportunity to present my research. And as Dr. Stat stated, today I'll be discussing one of my projects in the laboratory, which involves investigating the mechanisms of HIV-specific IgG3 antibody effector function and protection against infection. I will first begin by briefly reviewing the correlates of infection risk elucidated from the RV144 vaccine trial, and then discuss recent studies conducted by our laboratory looking at the HIV subclass response that was elicited by RV144 versus VAX-03. I will then delineate proposed studies to investigate the mechanisms by which these antibodies of the IgG3 subclass may confer partial protection against HIV infection, and then discuss implications for vaccine design. So the TIE Phase 3 RV144 vaccine efficacy trial provided an estimated 31.2% efficacy against acquisition of HIV infection at 42 months post-vaccination. An immune correlates analysis demonstrated that V1, V2 IgG binding antibodies correlated with decreased risk of infection. And it was found in a secondary analysis that ADCC, in the presence of low IgA, was an inverse correlate of risk. This was further supported by immune data showing that there was immune pressure against viruses that matched the vaccine at position 169 of the V2 loop of GP120. And as no broadly neutralizing antibodies were induced by RV144, these data suggest that non-neutralizing antibodies may have played a role in conferring protection. So did RV144 elicit a different magnitude and quality of response in VAX-03? So in contrast to RV144, the VAX-03 clinical trial utilized the same protein boost as RV144 without the ALVAC prime and showed no protection. And this was despite the fact that there were higher vaccine-induced neutralizing antibody responses in RV144 versus VAX-03. And Ferrari et al. Demonstrated higher ADCC activity in VAX-03 versus RV144. So we then asked the question, could qualitative differences in the specificity and or subclass selection account for the divergent efficacy outcomes observed between the two trials? In studies conducted by Nicole Yates and others of the laboratory, it was found that RV144 induced higher OMV IgG3, but lower OMV IgG1, G2, and G4 response rates in RV144 versus VAX-03. And interestingly, V1, V2 IgG3 responses differed between RV144 and VAX-03 and significantly correlated with decreased risk of infection. This was specifically seen for IgG3 antibodies that were specific for the V1, V2 sequence, matching major circulating strains in Thailand at a known site of immune pressure, GP70 V1, V2 169K, and this also correlated with decreased risk of infection. When we examine the structural and biological properties of the human IgG subclasses, IgG3 has the highest molecular weight of the secreted form as opposed to the other subclasses. And IgG1 is the most prevalent in adult serum, whereas IgG3 is found at low concentrations and has the shortest biological half-life. It is known that IgG3 is the most effective at activating complement and is capable of binding to all of the Fc gamma receptors, whereas IgG2 and IgG4 only bind to select receptors. It has been proposed that IgG3 has the highest function of the four subclasses due to the fact that it has the longest, most flexible hinge region of the subclasses. And from this diagram, you can see that the hinge region resides between the antibody variable region, or FV domain, containing antigen binding sites that are important for neutralization, and the FC, or constant domain, that's responsible for recruiting or engaging cells of the innate immune system to carry out antibody-dependent cellular cytotoxicity and phagocytosis. It is also known that IgG3 antibodies have the highest number of amino acids and inter-heavy chain disulfide bonds within the hinge region and are the most susceptible to proteolytic enzyme cleavage. There have been a number of studies looking at the role of IgG3 antibodies in immune-mediated pathogen control. They have been associated with long-term control of P. falciparum malaria and also the early appearance of neutralizing IgG3 antibodies correlated with chikungunya virus clearance and long-term clinical protection. When we look at HIV, it's been shown that IgG3 from polyclonal HIV immune globulin is more potent than other subclasses at neutralizing the virus. So based on the compelling findings that were generated in our laboratory showing that HIV-specific IgG3 distinguished the two efficacy studies and correlated with decreased risk of infection, and also data that historically IgG3 antibodies are associated with a more functional response, likely due to the long flexible hinge region, we formulated the following hypotheses. We hypothesize that IgG3 represents a mechanistic correlate of risk. It is a superior subclass response comprising multiple effector functions acting in concert or at multiple stages at the portal of entry. And alternatively, it may well be that IgG3 is a non-mechanistic or surrogate marker for another immune response that actually mechanistically protects. So the purpose of this study was to determine the antiviral functions that are improved with an antibody of the IgG3 subclass compared to IgG1, but having the same antigen specificity. These studies are significant in that the nature of the protective role of vaccine-elicited anti-IgG3 antibodies is not well understood. And while studies have been done to look at the magnitude and neutralization capacity of those antibodies, we still don't understand the specific Fc effector mechanisms that may have contributed to this decreased risk. Additionally, we don't know if this is a mechanistic or non-mechanistic correlative protection, or whether it's a correlate of risk or whether it's even a correlate of protection. These studies will clearly define the antiviral properties of a range of epitope-specific IgG3 and IgG1 antibodies and will serve as a benchmark for evaluating HIV efficacy studies. So to evaluate the antiviral functions that are improved with an antibody of the IgG3 subclass compared to IgG1, we used recombinant antibody technology to generate monoclonal antibodies of both subclasses. These antibodies were specific for various regions of the HIV-1 envelope, including V1, V2, V3, C1, GP41, and the CD4 binding site. We then compared the ability of the IgG3 and IgG1 monoclonal antibodies of the same specificity to mediate neutralizing and non-neutralizing antiviral functions. Monoclonal antibodies were isolated from vaccine recipients and HIV-infected donors and were produced by were originally also isolated by our B-cell team of investigators. We produced antibodies that were V2 specific from RV144 vaccine recipients known as CH58, CH59, HG107, and HG118. We also produced C1-specific antibodies that were known to mediate ADCC. And again, this was correlated with decreased risk of infection in the presence of low IgA. We produced the CD4 binding site antibody known as CH31 that is broadly neutralizing, 2 GP41-specific antibodies that are known to neutralize tier 2 viruses, in addition to a panel of GP41-specific antibodies that are relevant to ongoing vaccine trials. These antibodies were produced by transient transfection of heavy and light chain genes into 293F cells for production at the milligram scale. These antibodies were produced in 3 different backbones. We made IgG1 wild-type antibodies in the SEK backbone. We also produced IgG1 antibodies that have a series of alanine mutations within the IgG1 constant region that enhance Fc receptor binding in ADCC, and also produced IgG3 wild-type antibodies. We were first interested in assessing the binding and avidity characteristics of these antibodies, and we did this by surface plasmon resonance. We first focused on the V1, V2 specific antibodies and looked at their ability to bind to vaccine strain GP120s. The protein that was identified in the correlates analysis, in addition to V1, V2s that discriminate V1, V2 bNAbs, and the GP120 protein that induced higher IgG3 in RV144 versus FAXO3. And so for the SPR analysis, sensor chips were immobilized with the monoclonal antibodies of interest. Antigens were flowed over the antibody surfaces for a period of 5 minutes. Dissociation was followed for 500 seconds after on-protein injection, and then we measured the association, dissociation, and apparent affinity of these antibodies. This is showing you the time course of the binding of the V2 specific monoclonal antibody, CH58, which is a wild-type IgG1 antibody, the 4A version, and then the IgG3 version, binding to the GP70 V1V2 169K protein. And again, this is specific for a known site of immune pressure that correlated with decreased risk of infection. And we found that there were no significant differences in the binding affinity for these antibodies or the association or dissociation constant rates. However, I'd like to point out that we've only used OMF proteins that have been optimized for V2 exposure, and so in future studies we will look at the binding of these antibodies to proteins that are less well exposed to see if IgG3 antibodies can better bind those envelopes. So in addition to neutralization, antibodies are able to carry out a suite of effector functions, including virion capture, phagocytosis, and ADCC. For the neutralization assays, to compare the function of IgG3 and IgG1 antibodies, we wanted to look at their ability to neutralize tier 1 and tier 2 viruses. And for this, we utilized 3 different assays: the TZMBL-based, A3R5-based, and PBMC-based assays. We're interested in the ability of these antibodies to neutralize clade AE viruses, including a transmitted founder virus known as 427299. And these viruses were selected based on their sensitivity to RV144 plasmas. So in this graph, we can see that we compared the ability of CH58 IgG3 to neutralize the tier 1 AE virus 92TH023, and we found that this antibody was able to more efficiently neutralize this virus compared to the G1 variants, and we can see that that is apparent with the 2 to 3-fold decrease in the IC50 titer. So the lower the IC50 titer, the more effective the neutralization. We also looked at the ability of these antibodies to mediate infectious virion capture. For these assays, antibody virion immune complexes were formed in vitro, followed by absorption by the protein G column, infection of TZMBL cells with a flow-through fraction, and measurement of viral RNA by real-time PCR. These results are showing you the percent virion capture on the y-axis, and you can see the total virion capture as denoted by the black bars and infectious virion capture as noted by the gray bars. For the positive control, we utilized the monoclonal antibody PG9, which is able to capture both total virions and preferentially captures infectious virions. Our negative control was the Synagis antibody, in addition to RPMI alone, which show no virion capture. And consistent with published data, we found that CH58 captures low levels of infectious virions, and the IgG3 version of this antibody showed no enhanced ability to capture infectious viruses. So in addition to looking at those assays I previously discussed, we're interested in looking at the phagocytosis of IgG3 and IgG1 antibodies by looking at internalization of antibody-coated viruses and infected cells, utilizing fluorescently labeled viruses by flow cytometry and also image stream technology. And additionally, we will be looking at ADCC activity, measuring the ability of NK, monocytes, neutrophils, in mediating killing or destruction of target cells, and then subsequently measuring the release of cytotoxic molecules enabling apoptosis of target cells. So we expect that when our antibodies are produced in the IgG3 backbone, our data will show that OMV IgG3 is a mechanistic correlate of risk. And we think that all of these responses that I've discussed in the talk, neutralization, virion capture, ADCC, and phagocytosis, will contribute to an improved antibody functional response. We believe that these studies will greatly impact the HIV field in that we'll be able to better understand the functional profile of IgG3 antibodies against HIV. And this knowledge, we hope that we'll be able to leverage better vaccines, therapeutics, and diagnostics. So for future studies, we are interested in determining the possible synergistic interactions of IgG3 and IgG1 monoclonal antibodies for effector function, similar to what was done by Justin Pilara and colleagues. where they noted that V2 and C1 specific antibodies actually synergize for ADCC function. And finally, we're interested in looking at whether differential glycosylation impacts the inhibitory properties of IgG3 and IgG1 monoclonal antibodies. We will do this by looking at the function of natural or non-recombinant antibodies derived from EBV transformed cell cultures and examine purified IgG3 from RV144 and VAXO3 recipients. I would like to acknowledge my mentor, Dr. Georgia Tamaris, as well as Nicole Yates and others in the Tamaris Laboratory, as well as Dr. Barton Haynes, Dr. Larry Liao, who allowed me to produce the antibodies in his lab, Drs. Mattia Bonsignori and Dr. Kwang-Ki Wang, who will be doing the glycosylation studies with us, our collaborators, and the participants and clinical staff of the VAXO3 and RV144 vaccine trials. I'd also like to acknowledge our funding sources, the Bill and Melinda Gates Foundation, the NIH, the T32 Postdoctoral Training Grant, and the CFAR. Thank you for your attention.
We have time for a question. David?
Yes. Can you clarify what you mean by a mechanistic correlate protection and how the specific vaccine design that you're going to use addresses that?
So when I'm talking about mechanistic correlate, so we see that there is a correlation, but we don't really know the mechanisms that are actually contributing to it. And it may well be that this IgG is just a marker for another response. But really, to determine whether it's a mechanistic correlate of protection, there would have to be studies that would be done in non-human primates where we would identify an IgG3, IgG1 pair that had different function, and then infuse those antibodies in those non-human primates, and then measured whether the IgG1 or IgG3 antibody would be able to better protect. However, those studies have a lot of caveats in that we still don't really understand the differences between the Fc receptor function between non-human primates and humans, and we still don't know if the IgG3 from non-human primates acts in the same manner as that from humans.
Celia?
What's known about the difference in the immunological milieu that elicits the IgG1 or this one?
So as far as that data, I'm not particularly certain about that, but we know that possibly it could be the influence of the T helper cells in that they're able to provide better help to elicit an IgG3 antibody response. There have been some studies looking at that to show that. But yeah.
Thank you.
Thank you.